This is a session on nanofibers and their application in rotator cuff. I am joined here by Jed Johnson, which is the Chief Technology Officer of the Atrion, and we want to have a really nice discussion about the graft applications in rotator cuff. I'm Vahid Entezari from Cleveland Clinic. I'm the shoulder surgeon, and I want to introduce Jed. Jed, go ahead.
Jed Johnson, co-founder and chief technology officer for Atrion Orthopedics, and background in material science engineering from Ohio State.
Perfect. Thanks for joining us. And the main topic here is rotator cuff repair. The goal is, when we're doing rotator cuff repair, we want to promote the tendon healing, restore the function for the patient, improve long-term stability, and also give patients pain relief. Next slide. And the key is how we can improve healing. And this has been a holy grail of rotator cuff repair. We feel that we have addressed the patient's pain function, but this is an unaddressed, unmet need that we're going to improve with augments that we bringing to the mix. And today, Jed is going to educate me and us about how we started this and how the journey has been. And so the first question I have for you, Jed, is that if you can tell us what inspired you to develop rhodium, and what analogies from other developments of this nanofiber technology that led to development of this technology.
Yeah, and as Vahid mentioned, my background is tissue engineering, and throughout the years I've worked on some different things, from how we got started, which was glioblastoma research, looking at plastic cell culture plates. And so we were looking at how cells attach, migrate, and move on these cancer plates. And that's really forming the basis of how we can take fibers and put them into something like a vascular graft or a wound care product. And so as a company, we've expanded out into these different applications, really using synthetic fibers for the basis for all of this. And so taking that same scaffold that we can regenerate healthy tissue, like with the horse there, that was actually my dad and brother who operated on that horse, to something like a tissue engineered intestine, all the way to a rotator cuff. And so using a synthetic scaffold or a nanofiber scaffold to achieve that.
Yeah, I see a wide array of applications. How did we ended up focusing on rotator cuff healing?
So we, as you mentioned, we want to try to focus on biology of the rotator cuff and not just a mechanical augment. And so when we look at how the cuffs have not healed with how the technology is advancing, we want to try to influence that biology, that healing. And so we started with something that's synthetic. It's fully bioresorbable. We don't want something sticking in there to cause inflammation over the long term. We want something that will mimic that native ACM, and the nanofibers mimic the same size as extracellular matrix as what's found in there. And then we're focusing on that interpositional position. And so we really have this challenge of balancing strength and healing, and we're doing that really with microstructure of the graft and then the biologic environment that we're influencing. And so we can take this resorption of the graft and allow that to influence the biology. We can take the fibers and have it promote cell adhesion, cell proliferation, and then we can have native remodeling in that rotator cuff environment. And ultimately, that's what we're doing, is regenerating healthy tissue.
Yeah, that's actually, you're tackling the healing problem from different aspects. So
correct. Yep.
When you hone into the microstructure of the rhodium, what can you tell us about what this has in common with the extracellular matrix and why cells like to attach to this structure?
And it's a great question, and to me that's really the hallmark of the nanofiber technology and rhodium, is these nanofibers are mimicking the fibers found throughout your body. We call it extracellular matrix. And this video is showing, in this case this is whole blood, but it could be PRP, it could be BMAC, it could be anything. And the scaffold is wicking and holding that there into that position. And the fiber diameter from 20 years of nanofibers and publications and patents are all tailored to help these grafts grow cells, regenerate healthy tissue. And then this polymer composition, and we'll get into this a little bit, but it's a biphasic polymer. So we have a PGA component and a PLCL component that are breaking down, but the microstructure is carefully tuned to have a lot of porosity, about 85% porosity, and a fiber diameter that allows cells to truly wrap around and interact with the fibers.
Yeah, I mean, this is amazing when you think about, like, you know, the cells are coming from the patient, but you almost create a scaffold that mimics the, you know, the extracellular matrix, and the cells like seamlessly move from the patient's bone marrow to this new scaffold. So can you tell me a little bit about how cells interact with this extracellular matrix in that scaffold?
So we really designed it focusing on the microstructure, and in my material science background, we were really looking at how fast the materials resorb and how fast native tissue is remodeling. And so we see these typical healing cascades where you have inflammation, you have proliferation, and then you have a remodeling phase. And we can choreograph that with the material composition's breakdown. And so if we put something in there that breaks down within a day, you know, the tissue can't heal that quickly. And if we put something in there that might take years, you know, you're going to have that chronic inflammation going on and on and on. And actually, when I was going through school, and the tissue engineering concept was polymers that would last for 1 to 2 years, that's way too long. We need to really kind of, we call it guerrilla warfare, get in and get out, let that body do what it wants to do. So the PGA component is breaking down within a couple of weeks, and the PLCL component breaks down in a couple of months. And that biphasic nature is what is allowing cells to proliferate into there and regenerate that healthy tissue. And then these resorbable polymers are breaking down into glycolic acid, lactic acid, and caproic acid, which we actually have a lot of references to support the angiogenesis, VEGF production, a lot of pro-healing, pro-remodeling responses from those acidic byproducts.
Yeah, that's amazing. I mean, we always have this idea of like adding cells to our biology, to our repair, but always like the question I have is like, okay, I'm gonna throw the cells there. Are they staying? Are they just I'm gonna inject them and they're gonna leave the shoulder? So you're telling me this, first of all, this scaffold attracts cells, and also after that initial very critical few months, it just dissolves, disappears. So I don't have like a synthetic thing in my healing interface.
Correct. Yep. And in school, we were always taught tissue engineering. We were going to add cells, grow our tissue, you know, like the mouse with the ear growing on its back. And in reality, that's too complex to get through a regulatory pathway. And so what we're doing is doing what we might call an in situ tissue engineering or an endogenous tissue regeneration.
Yeah.
So we're using your body's biology and just providing a scaffold, a framework that your body can do that. And we're really focusing here on this enthesis because we want in this case, the emphasis on enthesis. If we see the tears are occurring there, that's really where we want our healing to occur. And so we're putting that scaffold there in between the bone tendon. There are some applications where we might want to put that on top of the tendon, or maybe both, one underneath and one on top. So it really depends on the patient, but our core focus, our core product was really helping that bone tendon reintegrate and that regeneration of those Sharpie fibers.
I mean, this interface was like, you know, almost like sacred. Nobody wanted to touch it, and kind of going in there and trying to potentiate the healing potential. I mean, there's something that, you know, it's unique to this product, right? And
you would not believe how many people we talk to when we say we're going to put a piece of plastic in between the bone tendon, and they just kind of look at us or laugh at us. And I don't know if we really got lucky or how we achieved such great results, but the study we ran at Colorado State showed significant improvement in strength. At 6 weeks, we were about 33% stronger, and out at 12 weeks, we were already out at 75% stronger compared to a standard rotator cuff repair without rhodium. And people are asking, you know, how can we do that? We've tried other things there in that inner position, and they actually made the strength of repair weaker. And one of the keys, we can look at the histology, and you can see this Sharpie fiber regeneration,
Yeah.
this restoration of that native enthesis, and we can show that histologically, but we can also see some insight from the mechanical testing. And so Dr. Tony Romeo was there during this testing, and the control scaffolds, the tendon would really just pop off of that shoulder. And when we repaired with rhodium, instead of the failure occurring in just a rapid pop, it actually tore, more like Velcro. So you could visibly see these physical entanglements, Sharpie fibers, re-bridging that gap.
Yeah, the quality of that work, like I will go one slide back, is that congratulations, you guys won the
Thank you.
Basic Science Award from ASCS, which kind of speaks to the quality of the work you have done. So when we go as a surgeon, I want to speak few words about how I got interested in this. So we have a clinical problem. We do the rotator cuff repair, the patient's pain goes away. Well, we kind of almost have given up on this idea of making the rotator cuff heal. So we know the high rate of like failure, and a lot of stuff that we do have some consequences, like putting grafts that patients react, have an immunologic reaction to it. So it makes us concerned about what are we going to change about our rotator cuff repair to achieve that goal of healing. And also, you don't want to break the bank. You don't want to add like thousands and thousands of dollars to a repair that, potentially, you have marginal benefit from augmentation. So that cost to benefit ratio is always, as a surgeon, comes into play. So my angle to this is like there is a real clinical problem, then there is a solution that it seems that it doesn't change the outcome, improves the outcome, adds more strength to the repair, and evident by multiple studies that are out there. This is one example. Brian Badman's group showed that you can reduce the failure rate of the rotator cuff to about 7%. There's another study that shows 9%. These are significant improvement because 35 to 50% is the healing failure that we see in run-of-the-mill rotator cuff repair that we kind of came to accept. And so if now you have a technology that can bring that to 7 to 9%, is a lot of like compounding effect of like many patients that potentially would heal that otherwise would have not healed their rotator cuff. So this is a quick video that I show Jed how I do this. Where I am, I treat a lot of like medium to large size tears. This is a very mobile tendon, as you can see, and because of the size of the tear, I have to put 3 anchors. Always I leave the OR as like, is this gonna heal or not? Now I have a way of preparing this graft, and again, it's simple and there's no extra steps. I pass it through my medial row anchors and slide into the shoulder. It's that easy. Doesn't add a lot of like, time to your repair. And because we have 3 anchors, you can see I have used 2 rhodiums between 3 anchors medially. You go forward and repair your tendon the same way that you do. It doesn't change a lot about your repair, and you go ahead and put this in a double row repair, and your tendon now covering the graft. Another application here is using essentially in an open application. If there's more than 50% of the rotator cuff in subscap tendons is tear, I can add this to my repair, put my medial row anchors, pass the medial row sutures through the rhodium, and here again I'm using 3 anchors medially and pass 2 rhodiums. And this speaks to your wicking ability of this graft. 2 touches of the tendon and it's completely saturated with the blood, and so you can go ahead and do a double row repair. So the bottom line is you can add it to your workflow as a repair of any rotator cuff tendon. You can go back and don't change anything in terms of technique. I don't need to learn a new technique. I know I can use it with any anchors, and there's no additional cost with disposable. And also the handling characteristics. I've used some of the grafts that are currently in the market. As soon as they touch water, they start disintegrating. So this is a graft that I can handle it in the shoulder. I can move it around, and so it actually stands the test of application in the shoulder. And
Talk about your trimming here, because you have a custom Vahid shape that you're implanting
So,
there.
I mean, I guess that speaks to versatility of this graft. Like you can make it for a small tear, and then you have a three anchor tear, you can actually trim it to apply it there. And also, you can work with this shape of the graft so that you can essentially not have it stick into the shoulder and not stick it out of your repair and truly cover the footprint.
So you can skew it medially, laterally, place it wherever you want on that
it's
footprint.
like in this video, we have it like, in a square shape. I cut it because I know how it's gonna sit in the shoulder. And by passing that medial row anchors, I know it's gonna nicely get spread over the footprint and it's not bunching up. And you need to kind of do some math as like what the distances are, but I would say if you create punch holes, then you can have this nice spread of the graft over the footprint.
Great.
So, and again, going back to these questions of do we need to kind of go with the market, like, there's all these myths that are around. I'll throw this at you. Do you need collagen in any augment that you put in the shoulder?
So our body is entirely collagen. So the amount of collagen that we would put into that graft is trivial. So my answer would be no, collagen is not necessary. And we're purpose about that because we're using synthetic materials so that we can make a completely consistent product. So we'll make 1 million of these devices and every one of those is identical. The same mechanical characteristics, the same resorption profile, and people that have used allografts and xenografts know there's inconsistency there. Some of them take, some of them don't. And so we're really trying to produce a level playing field.
I agree. I mean, that eliminates completely this possible immunologic reaction. We know there are studies out there that you can have a billion nodular reaction to a bovine collagen, so that eliminates that risk factor completely.
Correct.
So you kind of spoke into why you went into the inner position, so I'm going to skip that because I think you had a good answer. In terms of fiber alignment, talk to me, from a engineering standpoint, do I need to have fibers aligned? Are they aligned in the body? And why that matters or doesn't matter?
Yeah, it's a great point, and I've got a lot of publications on aligned fibers and their effect on cell migration, on cell alignment. So a lot of people would assume we have this natural alignment. You can see it there in the Sharpie fibers, so why not use aligned fibers? And the truth of the matter is, if we were putting this in a Petri dish, we would use aligned fibers. But because we're putting it in the body, and if you remember back on that healing cascade, we have this remodeling phase, and that remodeling phase is truly remodeling. It's eating up everything we put in and putting down a new material. Okay. So we could put in aligned fiber, it would all get eaten up and remodeled and deposited, and so we're really relying on the body to provide those physical cues to make that alignment, not the scaffold.
So maybe that's not like, core, and your clinical data and the, preclinical data proves that cells don't, differentiate, and that's the best area they want to go into.
Correct.
So I would say, to just wrap up, we place this graft interpositionally. It's synthetic. There is no added collagen in it. And also, because this scaffold is mimicking the extracellular matrix, all the cells that are in the bone marrow like to come out and populate this area and improve the healing. Right. Would you like to add, like?
And that scaffold there is serving as a wick to hold the biology where we want it. So it's designed to be there.
Perfect. So I will open it up if there's any question here or after this session. We'll be happy to answer your questions. Thank you.